the class of okay. Okay, so good morning, everyone. So thank you again for coming to this class. So the title of my class is a Microscope and Fluorescence Imaging for Understanding of the Small World. So the main theme of my class is seeing is believing. And last time, I talked about the history of the microscope and molecular imaging for understanding of the small world. So now you understand why it is important to know the small world and how humans have known the small world by development of microscopes. And I also talked about the molecular imaging technique for selectively detection of our target in the small world. So today, I'm going to talk about the fluorescence imaging and its application in biological and medical research field. So there are two topics. One is what is the fluorescence imaging? And the second one is the power of chemistry in molecular imaging technique. So last time I heard you belong to the Department of Chemistry. So you may think the fluorescence imaging research field for biology. It is partially true, but the chemistry is also one of the central part in this field. So I want to explain how chemistry has contributed to this area. Okay. So initially, let's briefly review the last class. The resolution of our naked eye is about 0.1 millimeter. So we can see the structure of B, but it is difficult to see the plankton. Of course, we can see some small things are moving, but we cannot directly see the structure of plankton by our naked eyes. So humans have been developed the optical microscope and electron microscope for understanding the small world. So microscopes can provide the detailed information of the shape and the structure of the samples. And I also explained how important it is to selectively detect our target in the crude condition. So because microscope can only observe the shape and structure, it is difficult to selectively detect our target in crude conditions. So researchers have been developed the molecular imaging technique for better understanding of our specific target in small world. Okay, again, we want to selectively detect our target in crude conditions. So this illustration shows the intracellular conditions. Okay, so as you can see, there are many proteins here. Okay, this is a mitochondria, and mitochondria include many kind of proteins. And okay, and again, we want to detect this protein selectively. So for achieving this goal, the researchers have been developed the molecular imaging technique with a molecular probe. So molecular probe consists of ligand and detection moiety. Here is the ligand and here is the detection moiety. So this ligand can selectively interact with our target. And we detect this detection moiety. That's why this method is indirect method for detection of the target. And there are several modalities for detection. So for example, the fluorescence or PET and so on. And in this class, I will talk about the fluorescence. Okay, again, so let's think, before talking about the fluorescence, let's think again, why mango is orange. So now you know it. So this is because mango can absorb colors except orange. So which means mango include many organic compounds that absorb light. So some organic compounds absorb certain wavelengths of light. So especially when the compound have conjugation bond, the compound can absorb light. And without conjugation bond, the compound cannot absorb the light. And importantly, some organic compounds shows fluorescence after absorption of light. So for example, so this is a slide of the absorbance and showing fluorescence, and the organic compound can absorb the light and show fluorescence. So this compound called as fluorescent dye. For example, this compound named as anthracene shows a fluorescence upon irradiation of light. And without light, this compound shows no fluorescence. So this is explained by Jablonski diagram. So before irradiation of light, electron locate at ground state S0. So after irradiation of light, electron move to the excitation state. And thereafter, so there are several energy level like S1 and S2. And initially, electron moves to S1 state by internal conversion. And thereafter, the electron goes back to the ground state. Usually, electron releases energy as heat. However, in the case of some compounds, electron release energy as fluorescence. And the wavelength of light of the fluorescence is obtained by this equation. So importantly, this equation, this is this is the constant value, and E means the energy here, or like this. And importantly, the wavelength is inversely proportional to energy. That's why the wavelength of fluorescence is always shorter than absorbance. That's why, so this is a representative absorbance and fluorescence spectrum. And this is absorption spectra, and this is emission spectra. So the emission wavelength is always longer than absorbance, and this difference of the wavelength is named as Stokes shift. To date, a lot of fluorescent dye have been developed, and nowadays, almost all wavelengths in visible light is covered by these fluorescent probes. Because as I mentioned, the emission wavelength is inversely proportional to energy. So if the compound have longer conjugation bond, the compound shows longer fluorescence wavelengths. So for example, this compound shows longer fluorescence than this compound. So today, a lot of fluorescent dye have been developed and used for biological and medical science. So last time, I explained how to use this fluorescence imaging in biological research. Let's briefly review the last class. Okay, in text book, you can find many kind of this kind of illustration for explanation of cell signaling. As you can see, this illustration include many types of protein, and some you can see some proteins moves to another localized another place in the cell. So like this, the dynamics of protein are very crucial for their function. However, how to detect the dynamics of such target proteins? Of course, just by using the microscope, we cannot see the dynamics of the proteins. For this goal, so now you use of protein inside cells. So last time I explained about the immunofluorescence technique. In this technique, researchers use the antibody. So this is antibody is a protein that specifically interacts with the target protein inside the cell. So researchers use the antibody conjugated with fluorescent dye. And this antibody conjugated with fluorescent dye is added to the cell. And we can observe the fluorescence spot. In each spot, this antibody specifically interact with target proteins. So that's why we can detect where the protein is. So these are the example of immunofluorescence that I showed you the last time. As you can see, this protein localizes in mitochondria like this. On the other hand, this protein localized at the vesicle type of organelle called as peroxisome. So like this, by using the molecular imaging technique, you can clearly observe the localization of target protein inside cells. Okay. So however, there are crucial issues in this method. So for example, antibody actually cannot penetrate the cell membrane. So this is because cell has lipid layer on the cell surface. And antibody is relatively large and hydrophilic. That's why in usual case, in the case of live cell, this antibody cannot pass through the cell membrane. So that's why we need to fix the cell and remove the cell membrane. So in the case of fixed cell, I mean the cells that doesn't the cells that don't have the cell membrane. So in this case, the antibody can penetrate, can go inside the cell. However, the fixed cell means a dead cell. That's why we cannot conduct live cell imaging with antibody technique. So which means the dynamics of protein cannot be obtained directly. So researchers have developed the fluorescence imaging method for live cell imaging. So today I will introduce the fluorescence imaging for live cell imaging, and one is a protein-based method called as fluorescent protein. And the other is a small organic compound-based method called as fluorescent probe. Okay, so I initially talk about the fluorescent protein. So the fluorescent protein is a protein with beta barrel structure. It's like a barrel and has a fluorophore inside this barrel. And in fluorescence imaging, this fluorescent protein is genetically fused with target protein. And this fused protein is genetically expressed in cells. That's why we can conduct a live cell imaging of the target protein. So I will show you the movie of the live cell imaging. Okay, so like this, so you can, this is a live cell image of mitochondrial dynamics. And as you can see, oh, please wait. Oh, oh, no. Oh, no, please wait. Okay. Okay. So as you can see, the mitochondria is moving. The organelle is not, organelle is always moving. And this is expanded image or image. And you can see here and here maybe also. Okay, you can see the division of mitochondria upon stimulation. So like this, the organelle and the protein is always moving, and the live cell imaging can show when and where the biological phenomena occurs inside the cell. Okay, so I also want to show another movie for understanding of the importance of live cell imaging. So generally speaking, organelle always exist. So for example, for example, cell always has nucleus, right? However, there are some special membrane that transiently forms under stress conditions. The stress granule is an example of such organelle. And stress granule transiently forms under stress conditions. So that's why it's a reversible organelle. So this is a live cell imaging of the formation of stress granule. Here you can see the time after the stress. And okay, once again. So initially we cannot observe the stress granule. However, after 13 or 14 minutes later, after stimulation, we can see the circle. We can see the relatively large vesicle like this. So these are stress granule. So like this, live cell imaging enables us to understand the intracellular dynamics of protein and organelle. So importantly, we can understand when and where the biological phenomena occur. Okay, so I also want to talk about the history of the fluorescent proteins. So fluorescent proteins were mainly developed by these three researchers, Dr. Osamu Shimomura and Dr. Roger Chen and Dr. Martin Chalfie. Okay, so fluorescent protein was first discovered by Dr. Osamu Shimomura. It is known that the crystal jelly shows the green fluorescence. However, he was interested in this fluorescence and wanted to know which which protein shows the fluorescence. And he caught a lot of jelly with his family and isolated the protein showing a green fluorescence. So this was reported in 1962, but at this time, the detailed structure of the green fluorescent protein was not disclosed. Also, importantly, the researchers didn't have clear idea how to use this protein for bioimaging. So at this time, the green fluorescent protein was just a scientific discovery. However, in 1990s, Dr. Roger Chen first disclosed the structure of GFP, and he shows GFP has a beta barrel structure, and also GFP includes the fluorophore here. And he scientifically disclosed why GFP shows green fluorescence. Okay, the mechanism of the formation. Okay, so the mechanism of the formation of fluorophore is shown here. So inside the beta barrel structure, there are 3 amino acids, and this nitrogen in amide bond intramolecularly attack to this amide bond, and after cyclization, dehydration, and oxidation, this fluorophore forms. Okay, and this mechanism, this finding is very, very important because by changing the amino acid, we can change the fluorescence wavelength. Okay, I will explain later. And in the same age, Dr. Martin shows an idea that became a breakthrough in molecular imaging. He, for the first time, shows the availability of GFP as an expression marker. So he showed that GFP can be explained in various living things. For example, the E. coli and C. elegans, and also mammalian cells. So these 3 researchers won the Nobel Prize in 28. And by the way, do you know which Nobel Prize they won? So this is actually the chemistry. So this is very, very interesting. So because the Nobel Prize committee evaluates their research as chemistry. So from this, we can know how large field chemistry is. I also want to emphasize one more thing. The GFP was discovered in 1962. However, at this time, this finding was just a scientific finding, and the researchers have not did not have idea for the for using the GFP. And in 1960s, the researchers shows how to use this fluorescent probe for imaging. So even if the research is not considered as important at this time, the research may be a basic of breakthrough in the future. Okay. Okay, so nowadays, as I mentioned, various color of fluorescent proteins have been developed based on the discovery of Dr. Roger Chen. So as shown, okay, so as shown above, he discovers the mechanism of formation of fluorophore, and by changing the amino acid sequence, researchers have successfully developed a various color of fluorescent proteins. So this is very important because we can conduct multicolor imaging with these fluorescent probes, fluorescent proteins. So as you can see, so this is a nucleus and mitochondria and microtube. So we can detect multiple targets selectively at the same time. Okay, so now you know how fluorescent proteins are important in fluorescence imaging. So this technique can detect target protein inside cells. However, there are however, the small molecules also play important roles inside cell. So, can fluorescent protein only detect proteins, or can fluorescent protein detect small molecules? Okay, the answer is yes. So this tool is called as biosensor, which consists, biosensor consists of two parts. One is a sensing domain and fluorescent protein. And the sensing domain can specifically bind with analyte. And after binding with analyte results in fluorescence change. So the mechanism of the fluorescence change is a bit complicated, so you do not need to understand the mechanism. However, by simply after binding with analyte resulting in fluorescence change. So here is an example of fluorescence imaging of analyte inside the cells. So after stimulation, the concentration of analyte change, so which causes a fluorescence change. So this is the calcium ion imaging in ER. So here you can see which kind of the stimulation, and actually the fluorescence change is not very large, but you can see the decrease of the fluorescence here. Okay, so like this. Okay, so this fluorescent protein-based sensor is a very useful tool for detection of small molecules, because by simply changing the fluorescent protein and sensing domain, we can detect the multiple target inside the cells. So in this paper, the authors developed the calcium ion sensor, cyclic AMP sensor, and ATP sensors. And they expressed these biosensors in a single cell at the same time and conduct the live cell imaging of these targets. So like this, fluorescent protein can be applicable for biosensing of analyte. Okay, so next, I will talk about the small organic compound-based sensor called as fluorescent probe. So the fundamental molecular design is the same as fluorescent protein-based sensor. This probe consists of fluorophore and sensing moiety, and binding with analyte results in fluorescence change. And actually, these fluorescent probes also can be applicable for detection of enzyme function, which means the activity of the target protein. So in this method, the fluorescent probe reacts with enzyme, resulting in the structural change and fluorescence change. I will explain the detail of this part later. I initially talk about the detection of analyte with fluorescent probes. Okay, so one of the most famous examples of fluorescent probe is calcium ion sensing. And calcium ion is very important ion for cell function, especially in cell signaling, because calcium ion plays as a second messenger. So Dr. Roger Chen, so he was deeply involved in the development of GFP. So he also developed this organic compound for for calcium imaging inside the cell. So this fluorescent compound called as FLA2 is consists of a binding site and fluorophore. So here is the binding site with calcium ion. So like this compound bind with the calcium ion, which causes a spectral change of fluorescence. So this is a spectral change of fluorescence, and with this probe, we can conduct the live cell imaging of calcium ion inside the cell. And importantly, as you can see, this cell shows a different fluorescence from here. I mean, after 35 minutes of the stimulation, this cell shows a stronger fluorescence than this cell. So like this, optical microscope can only can detect only the structure, the shape of the cell. However, fluorescent probe can detect can understand the heterogeneity of the cells. So actually, this cell has a different function from this cell. So the next important target is enzyme. Enzymes are protein that catalyzes a chemical reaction. So in this reaction, the enzyme generates a product by using substrate. And there are more than 500 enzymes in single cells. So you may hear the cell metabolism, such as glycolysis and TCA cycles. And importantly, the activity and expression level of certain enzymes largely change in diseases. For example, in the tumor cell, the glycolysis is enhanced in cancer cells for rapid cell proliferation. So that's why the tumor cells, in tumor cells, the proteins related in glycolysis is largely expressed than normal cells. So therefore, so the detection of target enzymes are very important for understanding of diseases. So this is a simple example of detection of enzyme activity with fluorescent probe. Fluorescent probe consists of substrate moiety and fluorophore. And enzyme removes the substrate moiety, which resulting in structural change of fluorescent probe. And this fluorescent probe shows a fluorescence. And as you can see, so this is a fluorescence. And as you can see, the fluorescence increases time-dependent manner and enzyme concentration-dependent manner. Of course, the fluorescence imaging of enzyme activity also can be achieved inside the cell. So this is a fluorescent probe, actually developed by myself, and this fluorescent probe can detect the activity of target enzyme in the cells. So for example, when we add the probe into the normal mouse, this fluorescence is like this. And however, in the case of the cells from disease model mouse, the fluorescence decreases. And after drug treatment, the fluorescence intensity enhanced. So like this, we can detect the enzyme activity in disease model with by using fluorescent probe. And okay, so this is an example of fluorescent detection of enzyme activity inside the cell. However, fluorescent probe can also be applicable in tissue. So in this paper, the authors developed a chemical fluorescent probe for PSMA. PSMA is an enzyme that highly expressed in cancer cells. And the authors isolated tumors from the real cancer patient. And as you can, and by just seeing a tumor, we cannot understand where the target protein is. However, by addition of fluorescent probe, we can clearly see the fluorescence like this. And we can understand, okay, so there are target proteins and there are no target proteins. So like this. So this is tumors by seeing by our naked eyes, and we can clearly understand the difference, and we can understand the power of the fluorescent probe. So now, so fluorescent probes are used for only research. However, in the future, the fluorescent probe may be applicable for clinical use, especially in operation. So let's imagine that in operation, we need to find cancer. However, it is not easy to distinguish the cancer from the normal cells. However, by using the fluorescent probe, we may be able to clearly detect the cancer cells like this. Okay, in this fluorescence, the fluorescence become on by cancer-specific enzyme. So like this, fluorescent probes may be able to visualize the cancers and eliminate cancers easily. So actually, so this is a plan for using fluorescent probes in clinical operation. So for example, in this figure, the doctor equips the goggle for detection of the fluorescence, and also the confocal and the microscopy may be also applicable for the operation. Okay, so indeed, researchers are developing the fluorescent probes for this goal. Again, so this is a fluorescent probe that react with cancer-specific protein. And this protein can be applicable for imaging the isolated tumors and the imaging in mouse brain. So here you can clearly understand where cancer is. So research for fluorescent detection of cancer during operation are now ongoing. Okay, so now you can understand the importance of fluorescent probes and how they are used in research. I also want to introduce example for using of fluorescent probe, especially in drug discovery. As I mentioned, cancer expresses the specific enzymes. For example, okay, this enzyme is very important for cancer cell because this enzyme generates a product which is important for disease. So for example, cancer proliferation, okay? And if a compound called as inhibitor can stop the function of enzyme, the cancer will die. So in this case, the inhibitor can be a drug candidate. However, how do we develop or discover such inhibitors? So high throughput screening is a powerful method for discovery of the inhibitor from compound library. So in this research field, a compound library that contains huge number of compounds. So for example, sometimes this compound library include more than 1 million millions of compounds. So and the researchers want to find drug candidate from this library. So in this assay, the researchers conduct high throughput screening and found a hit compound. And in high throughput screening, they conduct inhibition assay. Okay? However, we need to effectively conduct this inhibition assay because the inhibition assay need a lot of need longer time and mostly it's very cost. So for this goal, fluorescent probe can be used. So because the fluorescent probes show fluorescence after reaction with enzyme. So which means fluorescent probe can be a substrate of enzyme. And if the inhibitor can stop the function of enzyme, we cannot observe the fluorescence. So like this, and importantly, the fluorescence can be rapidly and easily detected with a plate reader like this. That's why fluorescent probes are useful tool in high throughput screening. Okay, so as I explained here, fluorescence imaging technique is a very powerful tool in biology and drug discovery. And in the future, the fluorescent probe may be applicable in clinical use. Okay, so I also want to the evolution of fluorescence imaging technique, so which is super resolution image. And in my class, I explained in the limitation of optical microscope. So the resolution of the optical microscope around 0.2 micrometer. So the resolution is the resolution is explained by this Rayleigh equation. And this equation includes the wavelength of light. That's why the resolution is dependent on wavelength of light. And last time, I explained the electron microscopy for overcoming this limitation. In this method, the electron is used for detection of target instead of light. So that's why electron microscope can provide the detailed structure of the sample. However, actually, the live cell imaging can't be performed with electron microscope. So that's why the researchers wanted to conduct the live cell fluorescence imaging, super resolution imaging with fluorescence. Okay, there are mainly 4 methods in fluorescence-based super resolution imaging. So state and palm and so on. And the principle of these methods are a bit complicated. So in this class, I will skip the explain of the detail of the mechanism. However, briefly, please understand that we can conduct supramolecular imaging by fluorescence. Also, it is very expensive. It's not a usual, not the common method, but still we can conduct it. We can conduct a super resolution image. So you can understand the powerfulness of the super resolution imaging by fluorescence. So this is the fluorescence imaging of mitochondria with the conventional confocal microscopy. However, however, the high resolution imaging can provide the detailed structure in mitochondria like this. And importantly, this imaging is conducted in live cells. So this is live cell imaging of super resolution imaging inside cells. So this is an organella, and So here you can see the structural change. So this is the expanded image of here. And like this, the fluorescence imaging also can be applicable for super resolution imaging in live cells. Okay, so this technique was developed by these three researchers. And here again, it is very interesting that they won the Nobel Prize of Chemistry. So you can understand how large field chemistry is. Okay, okay, so let's go back to the lake we initially saw in this class. So there are a lot of living things you can see by your naked eye. For example, the fish and insects. And now you know there are many living things that we can't see by our naked eyes. For example, the plankton. And you already know how important they are in this way. So that's why, so seeing small world is not just a hobby, but it is very important in science. Okay, and human develops microscopes for seeing small world. And this microscope have contributed in biological and medical research by providing the detailed information of structures. And, and by combination of microscope and molecular imaging technique, we can selectively detect our target in crude conditions. And these techniques have been used in biology, drug discovery, and the medical treatment. And importantly, the researchers and engineers in various fields have contributed to development of this technique for seeing the small world. So in the last 10 slides, okay, it's a bit earlier, that I predicted, but sorry, so I explained a bit earlier, but in the last 10 slides in my class, I want to talk about the power of chemistry in molecular image. Okay, so now you can understand the importance of molecular imaging, but you may think because you are you belonging to the department of chemistry, so the molecular imaging is technique in biology. Of course, it is true because the final goal of the molecular imaging is the discovery of biological phenomena. So you may have question, how does chemistry involved in molecular imaging? And actually, is chemistry need for molecular imaging? The answer is definitely yes. So the chemistry is always one of the central of molecular image. So in the last 10 slides, I want to explain this part. So the most important point is only chemists can design and synthesize the desired molecule. So the researcher, he's a Dr. Roger Chen, wanted to conduct the calcium imaging, and he designed and synthesized. Of course, the student might synthesize the probe itself, but anyway, he designed this molecule based on chemistry. And the biologist also can design and develop the sensors based on fluorescent protein, but the fluorescent probe, I mean the organic compound, also very, very important in molecular image. Okay, so for example, I talked about the flats in my previous slide. And this probe is very, very useful probe for molecular image, fluorescence imaging of calcium ion inside cells. And a lot of biologists use this probe. And for development of FLA2, organic chemistry is needed for design of this probe, organic chemistry. And coordination chemistry is important for the mechanism of binding with calcium ion. Because, okay, so last time a student asked me a question. So how to generate the selectivity of the probe? So selectivity of the probe is always very, very important. And the chemist designs a molecule based on chemistry and improves the selectivity by trial and error. So the knowledge of chemistry is very important for the design of molecule. And organic synthesis itself also needed for making this probe, and analytical chemistry also needed for measurement of the fluorescence. So like this, one probe is based on various fields of chemistry. Okay, okay, in this slide, I will explain the example of importance of chemistry in molecular design. So in fluorescence imaging, photobleaching is a very, very, very crucial issue. The photobleaching is a decomposition of fluorescence dye after irradiation of light. So for example, in museum, we are not allowed to take a picture. So this is because the strong light may decompose all the pigment. Okay, so this is the same in fluorescence imaging. When we use a too strong light, the fluorophore decompose by irradiation of light. So this is before the irradiation of strong light, and when the strong light is irradiated, this part, the fluorescence disappear. So this is a photobleaching. And this photobleaching is explained by this scheme. And after irradiation of light, compound becomes excited state. And usually this excited state is, okay, the electron go back to the ground state. However, some of the compound react with oxygen species, resulting in the decompose of the fluorescent dye. So this is another case of the photobleaching by irradiation of light. So anyway, the excited fluorophore react with oxygen species. So to overcome this limitation, the chemist developed fluorescence dyes with high photostability. So in this paper, the authors introduced the electron withdrawing group. So the density of electron decreased like this. And in the case of the conventional fluorescent dye or this type, the fluorescence decreased by irradiation of light. However, in this probe still shows a strong fluorescence after irradiation of strong light. So that's why. So this probe can be applicable for long time in imaging. So in the case of this fluorescent dye, we can still observe the strong fluorescence after irradiation of light. However, in the case of usual fluorescent dye, we cannot observe the fluorescence because of the fluorescence bleaching. So like this, chemists can design and synthesize the desired molecule based on chemistry. Okay, so chemistry is also used in metal ion sensing. So in my previous slide, I explained the calcium ion sensing with this probe. And this part is the binding moiety with calcium ion. And by simply changing the binding site, we can detect other metal ion like this. So coordination chemistry is also used for metal ion sensing. And organic synthesis is also very important for development of probe. Okay, so last time I explained PET imaging. In PET imaging, radioactive isotope spontaneously decompose and release positron. And this positron react with electron, resulting in release of gamma ray. And we detect this gamma ray. However, the important point is this spontaneous decomposition. So generally speaking, half-life time of PET probe is very short. So in the case of this probe, the half-life time is only is about 2 hours. So after several hours, we cannot use this probe as a probe. We cannot use this compound as probe. So which means chemists need to rapidly synthesize a PET probe, including purification. I show an example of rapid synthesis of the complicated probe by organic chemists. The PET probe can chase the dynamics of drug in our body. So, for example, in the case of this anti-cancer drug, okay, so we want to know the localization of this probe. And the PET imaging is very useful approach for this goal. And when we use this PET probe, okay, this PET probe has the radioactive isotope here, we can directly observe the localization of this drug. However, the half-life time of PET probe is very, very short, and only actually 20 minutes. So chemists developed a rapid synthesis method for this probe. Okay, again, the half-life time of the carbon-11 is only 20 minutes. And there are several methods to introduce a carbon-11 to this probe. And in this paper, the authors developed a new synthetic method for nitroaldol reaction to introduce the carbon-11 here. So this, okay, and this is a synthetic route for PET probe. As you can see here, the time for each step are very short, and the authors successfully synthesized this probe within 20 minutes, surprisingly including the HPLC purification. So after synthesis, they quickly administrated this probe and conduct the PET imaging. So like this, the organic synthesis chemists have also contributed to the development of molecular probe. Okay, so now you can understand how chemistry is important in molecular image. But you may think, actually, I'm interested in inorganic chemistry rather than organic chemistry. So of course, the inorganic chemistry is also very important in molecular image. So for example, the quantum dot. So this is a metal-based nanoparticle that shows a fluorescence. And the quantum dot has a core shell type structure and shows fluorescence. And importantly, the fluorescence wavelength depends on its size. And this quantum dot have this quantum dot has good photostability and bioavailability. So the quantum dot is conjugated with antibody, and this probe is used in bioimaging. And again, so this quantum dot was developed by 3 researchers, and they won the Nobel Prize in Chemistry last year. Okay, and surprisingly, inorganic chemists use the diamond for fluorescence imaging. So nano diamond also has high photostability and high bioavailability. So also the diamond is, of course, more expensive than the organic compounds anyway. So diamond, even diamond, can be applicable for fluorescence image. So the useful probes for fluorescence imaging have been developed based on inorganic chemistry. Okay, so as I mentioned, various fields in chemistry have contributed to molecular imaging and better understanding of small world and our better life. So the organic chemistry is needed to molecular design and synthesis itself. And inorganic chemistry also provides useful tools. And analytical chemistry also needed for the measurement of the fluorescence. And the physical chemistry also needed for the basis of theory. So you can also understand the importance of the chemistry by the fact that Nobel Prize of Chemistry is awarded to the 3 research topics in molecular imaging. So like this, the chemistry also one of the central research field in molecular imaging. Okay, so it's a bit earlier, but sorry, but this is the last slide in my class, and this is my personal message to you as a chemist. So now you learned chemistry in your department, and after several years, or now for some students, you will or now you do the chemical research in a laboratory and will get a master and doctor degree. So you are here, and after this, you may be the expert in chemistry. So this is an excellent pathway, and please do your best to be an excellent chemistry. And you can also go to other field in other research field like me and Dr. Samir also. And however, what I want you to know is, no matter which research field you will go, the chemistry will be always your basis. So for example, nowadays, the fusion research is very, very important. So biologists may ask you to synthesize or design the molecule. And chemists also may ask the biologists to evaluate their probe. So like this, fusion research is very important. But please remember that the chemistry will be always your basis. So my message is learn chemistry hard and do experiment hard. Okay, and I also recommend you should play hard before going to a laboratory, because we will not have enough time for playing after belonging to a laboratory. Okay, and I also want to recommend that and if possible, I also want you to get a doctor degree. This is a license as a researcher for global activity. So okay, if there are no financial issues, I really recommend to get a doctor degree. Okay, so it is a bit earlier, but this is my last source. Okay, thank you very much for joining this class. Okay, that's all. Thank you.
Thank you so much, Dr. Shohei Uchinomiya. Super comprehensive lecture.
Yeah.
I really enjoy it. I feel like I go back to my days during PhD, listening to your lectures.
Yeah.
It's
Yeah.
our kind of daily activities, right?
Yes,
Our
yes.
period in Singapore.
Yes, true. I remember. Yeah. I also
Yeah.
remember.
Yeah, I kind of have flashback just now. Thank you so much. If you don't mind, do you still have time to take questions? Probably.
Yeah, of course,
If
of course.
anybody from the floor, if you have any questions.
So any kind of question is okay. So for example, why we need to get a doctor degree, or what we should do in laboratory or other things. So the question is not limited in the research. Research, I mean the molecular imaging. So
Maybe any of you interested in getting master or doctoral degree in Kyushu University in Japan?
Yeah. It's also you are always welcomed. So as I mentioned, getting the master and especially a doctor degree is really important for global activity. I think, I believe so. So getting the doctoral degree in your university is also very important. And if you have a chance, go abroad. So like Samira, and you can also go abroad as a postdoc. Yeah. You as a postdoc like me, and it will be a great experience for you.
Maybe Dr. Shohei, to also, maybe you can share a little bit about your degree journey. Like after you get your degree, PhD degree, and then you did your postdoc, and then what happened after that?
Oh,
You can inspire us.
Okay. Yeah, of course. But
if you don't mind.
yeah, of course. But what should I talk? Which topic should I talk about? Oh, okay. The one thing is so my experience in Singapore, right? So the most important thing is actually we can conduct the scientific research in Japan. So if we just want to do the research, I might not need to go abroad, because there are many good research institutes in Japan. But I in Singapore, I had a great experience with my colleagues. So, for example, Dr. Samira also, and other researchers in Professor Chan's in my laboratory. So I was, how can I say, I can get many stimulation from them. So yeah, uh, it may be a bit difficult to explain how to say English, but anyway, you can understand my feeling if you go abroad. So you can also ask Dr. Samira about her experience, because she got her PhD degree, right? Not postdoc. So she chose, she selected her pathway. So for getting the PhD degree in Singapore. So she is a very good example, I think.
Oh, yeah.
Yeah.
You are too nice. You are too kind.
No. It's a rarely. So, uh, actually, even if you will go to industry, so I mean, the not academic research area, you have many chance to go abroad. So, for example, my brother and my friends also go abroad, so for 1 or 2 years. So, I so this age is a really global age. So, for being a research for how can I say that? To be active researcher, to be active as a researcher, we need to get a PhD degree, and we also need to improve our English in our English. So actually, when I first went to Singapore, my boss is a Professor Chan, strictly told me to improve my English. So I really recommend you to learn English now. So yeah, before going to abroad. Yeah.
Oh, by the way, related to that, have I told you in December last
Well,
year, Prof. Chang came to my department.
oh, really?
Yeah,
Oh,
have I told
great.
you? I haven't?
Oh, no,
Okay.
I haven't
Yeah,
known that.
he
Oh, okay.
also gave a guest lecture, but
Oh, okay.
offline. So he physically came here and see around the campus, and
Oh,
he
great.
visited my office.
Oh, great. Oh, great.
Yeah, so I believe some of the students here that attended this lecture, they have met Prof. Chang Yangti before. I think I see some names here. Because they helped me to organize the lecture, and then they got a chance to have lunch together with Prof. Chang. So
Great.
yeah, it's a very nice having Prof. Chang and having you in my department. So I really hope this relationship, I would say, this
Oh,
conversation will go further with you, with Prof. Chang, and all of our alumni from our previous lab.
yeah. It was really nice. Yeah. So, the Professor Chan is also a very good chemist. So his base is also chemist. Okay. So, so I'm not sure which kind of chemistry you are learning, but please study chemistry hard. So chemistry is always your very big weapon. Yeah, I think so. I believe so. Okay, so
Maybe
now it's almost 11:50 in your country, right?
yes, correct.
Okay, so uh, uh, I think students want to have lunch now, because cafeteria also will be crowded. So,
Exactly.
it's okay to finish a bit earlier.
Yeah. Oh, okay. Before we close this session, could we please have a picture together? Like
Oh,
everyone. I think I have here Hassan. Hassan, can you help me to take a picture? And everyone, all students, the attendees, could you please on your camera so we can capture this beautiful moment?
okay. So I also want to take a picture. Yeah, okay.
Everyone, you can on your camera if that's possible. Okay, maybe okay, they need to, make tidy up their hair and put on their stuff, the clothes and everything. So it takes time for them to on the camera
It's
probably.
okay, it's okay.
It's usually like that, right? If you have a
Mm.
lecture online, and then whenever I ask them to, can you on your camera? And then they will be busy like, oh, my hair and this and that.
Yeah. True.
Yeah, the typical of us. Hassan, are you there?
Yes, Miss Amira.
Okay, Hassan.
Okay, I will be taking pictures. There will be 2 slides, so I'll be counting down for until 3 2 times. I think I will wait 1 more minute for everyone to turn on their camera.
Can you see more people on their camera?
No, it's okay. It's okay.
All right.
Okay, for the first slide, I'll be taking 1, 2. Okay, for the next slide, I will be taking 1, 2. And there is another slide for Dr. Uchinomiya Sensei. There's a certificate from our department for Dr. Uchinomiya. Wait, let me share one more slide.
Share one slide. Okay. Please wait.
So it's kind of our little bit token of appreciation that we can do online because we are too far away in distance.
Okay.
So
Please wait.
the department prepared this kind of certificate
Oh.
for your kind willingness to share with us. Is it?
So I share
No,
the slide, no?
no. I think Hassan will share. Hassan, you want to share a screen now?
Yes, it's on progress.
Okay, sure.
Is it visible?
Yes, I can see now.
Okay, so thank you so much for
Oh, thank you.
Dr.
Yeah.
Shohei Uchinomiya Sensei. And I will be taking picture one more time together with all the audience. Okay.
Sure.
1, 2. Okay, thank you so much. I will be giving back to
Thank
Miss
you.
Samira.
Yeah. Thank you so much, Dr. Shohei. So apparently, unfortunately, our HOD, head of department, cannot make it today to convey the message to you. So he actually wanted to kind of give this token of appreciation to you, but he has other schedules. So again, I would like to say thank you so much on behalf of department and our university for you to join us in this and give us these two very comprehensive and wonderful lectures. I hope it can be a start for us to collaborate more in the future between University of Indonesia, especially our Department of Chemistry, and also your department in Kyushu University.
Yeah.
I hope we can continue our collaboration further. So if you need anything, if you want to come to department, please just let me know. We will arrange everything for you.
Okay, thank you very much. Yeah, I also want to visit your office.
Okay, in that, I want to say thank you to all the audience and have a good day, everyone.
Okay.
Bye-bye, Dr. Shohei Uchinomiya.
Okay, thank you very much. Thank you.
Bye-bye.